Lung cancer is a type of cancer that begins in the lungs. Your lungs are two spongy organs in your chest that take in oxygen when you inhale and release carbon dioxide when you exhale. Lung cancer is the leading cause of cancer deaths worldwide. The diagnostic, which requires only a simple urine test to read the results, could make lung cancer screening more accessible worldwide. Using a new technology developed at Massachusetts Institute of Technology (MIT), diagnosing lung cancer could become as easy as inhaling nanoparticle sensors and then taking a urine test that reveals whether a tumor is present. The new diagnostic is based on nanosensors that can be delivered by an inhaler or a nebulizer. If the sensors encounter cancer-linked proteins in the lungs, they produce a signal that accumulates in the urine, where it can be detected with a simple paper test strip. This approach could potentially replace or supplement the current gold standard for diagnosing lung cancer, low-dose computed tomography (CT). It could have an especially significant impact in low and middle-income countries that don't have widespread availability of CT scanners. Around the world, cancer is going to become more and more prevalent in low and middle-income countries. The epidemiology of lung cancer globally is that it's driven by pollution and smoking. So we know that those are settings where accessibility to this kind of technology could have a big impact. To help diagnose lung cancer as early as possible, the US Preventive Services Task Force recommends that heavy smokers over the age of 50 undergo annual CT scans. However, not everyone in this target group receives these scans, and the high false positive rate of the scans can lead to unnecessary invasive tests. In present study, scientists explored the possibility of using nanosensors as a more accessible alternative to CT screening for lung cancer. These sensors consist of polymer nanoparticles coated with a reporter, such as a DNA barcode, that is cleaved from the particle when the sensor encounters enzymes called proteases, which are often overactive in tumors. Those reporters eventually accumulate in the urine and are excreted from the body. Previous versions of the sensors, which targeted other cancer sites, such as the liver and ovaries, were designed to be given intravenously. For lung cancer diagnosis, the researchers wanted to create a version that could be inhaled, which could make it easier to deploy in lower resource settings. To achieve that, the researchers created two formulations of their particles: a solution that can be aerosolized and delivered with a nebulizer, and a dry powder that can be delivered using an inhaler. Once the particles reach the lungs, they are absorbed into the tissue, where they encounter any proteases that may be present. Human cells can express hundreds of different proteases, and some of them are overactive in tumors, where they help cancer cells to escape their original locations by cutting through proteins of the extracellular matrix. These cancerous proteases cleave DNA barcodes from the sensors, allowing the barcodes to circulate in the bloodstream until they are excreted in the urine. In the earlier versions of this technology, the researchers used mass spectrometry to analyze the urine sample and detect DNA barcodes. However, mass spectrometry requires equipment that might not be available in low-resource areas. So for this version, the researchers created a lateral flow assay, which allows the barcodes to be detected using a paper test strip. The researchers designed the strip to detect up to 4 different DNA barcodes, each of which indicates the presence of a different protease. No pre-treatment or processing of the urine sample is required, and the results can be read about 20 minutes after the sample is obtained. The researchers tested their diagnostic system in mice that are genetically engineered to develop lung tumors similar to those seen in humans. The sensors were administered 7 weeks after the tumors started to form, a time point that would likely correlate with stage 1 or 2 cancer in humans. In their first set of experiments in the mice, the researchers measured the levels of 20 different sensors designed to detect different proteases. using a machine learning algorithm to analyze those results. The researchers identified a combination of just 4 sensors that was predicted to give accurate diagnostic results. They then tested that combination in the mouse model and found that it could accurately detect early-stage lung tumors. For use in humans, it's possible that more sensors might be needed to make an accurate diagnosis, but that could be achieved by using multiple paper strips, each of which detects 4 different DNA barcodes. The researchers now plan to analyze human biopsy samples to see if the sensor panels they are using would also work to detect human cancers. In the longer term, they hope to perform clinical trials in human patients in parts of the world where there is limited access to CT scanning. This technology could offer a dramatic improvement in lung cancer screening, especially since the results can be obtained during a single visit. The idea would be you come in and then you get an answer about whether you need a follow-up test or not, and patients can be identified who have early lesions into the system so that they could get curative surgery or life-saving medicines. Thank you for watching. If you've enjoyed our videos, subscribe to our channel, but there's more to come.